Today we're going to start Lecture 2 of Unit 5, and in this lecture we're going to be focusing on lipid digestion, absorption, and blood distribution. We're first going to discuss lipid digestion, focusing mainly on non-ruminants first because it's a little more clear-cut in non-ruminants than it is with ruminants. So our lipid digestion in non-ruminants involves the mouth, the stomach, and the small intestine. And it actually, for a lot of species, starts in the mouth. So there is some lingual lipase that is released. However, this is not the case in avian species. They do not have any lingual lipase. And there are discussions on, there's usually in mammals more lingual lipase in neonatal animals that are consuming milk, more so than adults. So adults may lose this lingual lipase. And another main component that's necessary for proper lipid digestion is we have to have emulsification of the fats. And emulsification is where we actually are dissolving fat in water. And that emulsification begins in the stomach. It does not begin in the mouth. But so all that churning of the stomach is really what's starting that emulsion process. And these lipases that are found both in the stomach and the mouth are what we refer to as acetic lipases. So these contribute about, or do about 30% of the hydrolysis of all the triglycerides that come in. And in neonates, it's about 50%. So overall, they're not contributing very significantly to lipid digestion, and some nutritionists will actually discount these enzymes on their overall contribution to lipid digestion. But I feel as though they are there. They're important for various stages of life, mainly our neonates, so we may as well discuss them. These do not require bile salts or co-lipase, and they have an optimal pH between 3 and 6, and they cleave at the SN3 position. And this SN3 position is actually the same as saying the third carbon. So they only cleave there. So we're first going to discuss a little bit of these lingual and gastric lipases, or acetic lipases. So our lingual lipase, lingual meaning tongue or mouth, so lingual lipase is secreted from the lingual serous gland, the von Ebner gland, and it specifically hydrolyzes medium and long chain triglycerides in the stomach, and we end up getting the result is one non-esterified fatty acid. This is also referred to in various texts as free fatty acid. But as you know, when we have our fatty acids conjugated to our glycerol molecule, that's called an ester bond. So these are non-esterified fatty acids. And then we get a diacylglyceride. That's what the DAG stands for, meaning that we have a glycerol and two fatty acids. And then we also have some species that have the gastric lipase. This gastric lipase is secreted from the chief cells, and it hydrolyzes medium chain triglycerides. So the fatty acids on the triglycerides that are medium chain. And there's a high level of gastric lipases within our raptors. So our avian species do have gastric lipase. And here on the left-hand side, we have a graph showing the concentration of lipases, so we have our lingual or our mouth and then our gastric. So you can see our rat and our mouse have high amounts of lingual lipase, and our rat basically has 0 gastric lipase, and our mouse just has this tiny, tiny, tiny amount that you can see there. Where if you look at other species, our rabbit, guinea pig, baboon, and human, they have a decent amount of gastric lipase, but really have negligible lingual lipases. So it really varies on the species on if they have lingual or gastric, and very rarely are you having a significant amount of lingual and gastric lipase occur in the same animal. Now here we have a immunohistochemistry diagram looking at gastric lipase in humans. So here you could see that our gastric lipase is co-localized with pepsinogen in the chief cells. So our pepsinogen is staining yellow, our gastric lipase is staining red. Yellow and red together make orange. So that's why all this orange is showing that our gastric lipase and our pepsinogen are all within the same chief cells, and they are localized together. So why do we have these lipases, the lingual lipase and the gastric lipase? I would say that even though the lingual lipase is secreted in the saliva, it's actually doing its action in the stomach. So it's moving along. So they both are having similar action. And that also makes it difficult if you're trying to determine if it's lingual or gastric lipase within the stomach that's doing the main action. So these lipases in the stomach, they are critical for adequate lipid digestion in our neonates. So our neonates have immature pancreatic lipase, so their lipases are not very adequate at breaking down. They're not making adequate amounts of pancreatic lipase. So it's very critical to have these lingual and gastric lipases to break down the fat. It has the greatest activity on milk fat, and that's mainly due to the amount of those medium chain fatty acids that the gastric and lingual lipase prefer. And then their products, they are also acting as surfactants. So these surfactants, that's basically, surfactant is similar to soap. A soap is a surfactant. And so that is allowing it to spontaneously absorb water at the liquid interface, and it stabilizes the emulsions. We have gastrin causes the release. So just as gastrin causes the release of pepsinogen from the chief cells, it makes sense since pepsinogen and gastric lipase are co-localized, that gastrin also causes the release of gastric lipase. And then we have secretin and CCK, cholecystokinin. They inhibit the release of the gastric lipase and the pepsinogen. Okay, when we leave the stomach, we then are reliant on our pancreatic lipases. So our lipids leave the stomach, they enter the duodenum, we get the release of CCK and secretin that's going to cause the pancreas to release its gastric juices. So in addition to all the enzymes that the pancreas is releasing to digest proteins, it's also releasing lipases to help digest lipid. So there are 3 lipases. We have the first one is lipase, we have lipid esterase, and then we have colipase. So these are found in the acinar cells, in the zymogen granules, and they act at the water lipid interface. And we're going to get into the specifics of each one of these lipases in a few minutes. In addition to the lipases, we have phospholipases. And phospholipases are in the form of a zymogen. So they are pre-phospholipases. And the reason why we want to do this is we don't want to be breaking up our cell membrane unintentionally. So we have them in the zymogen form. They are activated by trypsin, and we have 5 types: A1, A2, B, C, and D. And we'll be discussing where those break in a few minutes. So the main component of our lipid digestion is primarily lipase. This is the main focus of our lipid digestion. And it cleaves at the SN1 and the SN3 position. So this would be carbon 1 and carbon 3. It leaves that second carbon, that middle carbon, alone. So therefore, we get 2 non-esterified fatty acids and 1 monoacylglyceride, or 1 MAG. So here we have our triglyceride. We have our 1, 2, 3 fatty acids on our glycerol backbone right here. When we have our pancreatic lipase, we are cleaving off number 1 and number 3. So we're getting 2 non-esterified fatty acids and 1 monoacylglyceride. Our lipase actually does not require bile salts for its action, and it's actually inhibited. Its action is inhibited by the bile salts. So why do we have bile salts? Well, that's what we need colipase for. So our colipase is stabilizing the lipase in the presence of the bile salts, and our colipase require bile salts. So that's why all of that, why we have that. So what is our colipase? Our colipase is a small protein. It binds to the water lipid interface and lipase, and basically it anchors and activates the lipase. So we need the colipase for the lipase to work adequately. And next we have our lipid esterase. So lipid esterase also requires bile salts. So this is where lipase has to compromise. It doesn't like the bile salts, but there are other enzymes that require bile salts. So that's why lipase utilizes colipase. So what does lipid esterase do? Lipid esterase cleaves the fatty acids from cholesterol esters. Okay, our last classification of the enzymes are phospholipases. And our phospholipases also require bile acids or bile salts for activity. And they are differentiated by the ester bond they hydrolyze. So as I said, there are multiple phospholipases. And so here we have our phospholipase A1. So this is cleaving at the SN1, or the first carbon. And then we have our phospholipase A2, that's at our second carbon. And I'd like to note that this second carbon, so our phospholipase A2, is the first one that is hydrolyzed. And our phospholipase C here is what is cleaving the phosphate group off of the third carbon where our phosphorus-containing molecule is. And then our phospholipase D is what is cleaving and taking the choline off of the phosphate. So that is the different phospholipases. I do want you to know what A1, A2, C, and D do and where they cleave. I have mentioned bile salts or bile acids a decent amount throughout this class so far, but we haven't really explained what bile acids are. So bile acids are synthesized by the liver in the peroxisomes, and we have primary bile acids, and this is synthesized from cholesterol. So as I mentioned in the previous lecture, that cholesterol is a precursor for our bile acids. It's a precursor for our primary bile acids or bile salts. And we have cholic and cyanidioxycholic acid. And both of these have a hydrophilic and hydrophobic end, and that's how they help make the emulsion. They are stored in the gallbladder, which we already knew, and they are secreted when cholecystokinin signals the release of the gallbladder. Now, when our bile acids do enter, especially the hindgut, we have intestinal bacteria, and these are able to modify the bile salts, and this forms the secondary bile acids. So our primary ones are the bile acids that we are able to synthesize. Our secondary bile acids is what has been modified, and that is referred to as secondary. So when I was talking during amino acids, specifically when we were talking about taurine, I mentioned that taurine is conjugated to bile salts. So our bile salts can be conjugated to either taurine or to glycine. Now, taurine is conjugated to the bile salts in birds, cats, and ruminants, where we utilize glucine in our hindgut fermenters. So this would be horses, rabbits, etc. And omnivores use a mixture of both taurine and glycine. So why do we have this difference? Well, glycine conjugates are less soluble in acidic environments. So that tells us that our birds, cats, and ruminants have a more acidic GI tract than our hindgut fermenters and our omnivores. So our glycine is less soluble in acidic environments. So we like a higher pH. Our glycine likes a higher pH. And as I mentioned before, we have the secondary bile salts or bile acids, and these are carcinogenic. So this is one of the reasons why individuals that don't eat adequate amounts of fiber, that consume large amounts of fat, red meats, etc., they are more prone to colon cancer. So the secondary bile salts, they actually damage the colonic epithelium, and at the same time, it stimulates crypt cell proliferation. So we have more of these cryptic cells proliferating, and we damage additional components of the colonic epithelium. All of that is going to lead to increased rates of colon cancer. So this is one of the reasons why it's important to eat adequate amounts of fiber to keep your bowel movements regular, because the longer you have your digesta stored within your colon, you're going to be subjected to those secondary bile salts or bile acids that the microbes have converted, and therefore you're at a greater risk of colon cancer. So the main function of these bile acids is that they help form these micelles. And a micelle is composed of the bile salts or acids and a phospholipid layer. So our phospholipids, we have our hydrophobic ends are out, which is our phosphate group. Our hydrophobic ends are in, and basically that forms this disc shape here. So a lot of times we think of these micelles as being little spheres, but they are actually discs. And the core of the micelle is filled with triglycerides, our non-esterified fatty acids, monoacylglycerides, our cholesterol, our fat-soluble vitamins, and any drugs that are hydrophobic. So this is one of the benefits that our pharmaceutical companies utilize. If you want longer lasting drugs, they will make them more hydrophobic, so then they can incorporate and they're slowly released. So here on the schematic, you can see the brown is the bile. We have our over here, so that component, the sides of the disc are made up of our bile, our phospholipids here, and in the center we have our other lipid components, our triglycerides, fatty acids, etc. So why do we want to form these micelles? Well, these micelles are assisting with the emulsification, and those emulsifications allow the pancreatic lipase to attach to the micelle and digest the lipids. So here we have our emulsified fat up here, and our enzymes able to bind to that. So our lipases and our phospholipases are now able to easily attach to the micelle and begin lipid digestion. So that's why we need to form those micelles. So how does lipid digestion differ in ruminants? Well, the natural diet of ruminants is very, very low in lipids. It really doesn't have that much lipid content. And the lipids that are there, we have galactolipids, phospholipids, a lot of waxes, pigments, and essential oils. These leaf lipids have very few triglycerides. Now, the triglycerides are mainly in the seeds, and that's because the seeds, when they germinate, are going to need access to those triglycerides for energy. So our triglycerides are coming from our seeds, our corn, soybean meal, etc. In addition, our microbes do not utilize lipids very efficiently. Within the rumen, they do not have an emulsifying agent. We don't have any bile salts released within the rumen, so that they don't have the assistance of that emulsification to help attach onto those triglycerides or lipid droplets to break them down. In addition, the unsaturated fats are toxic to the microbes. So because those polyunsaturated fatty acids are toxic, they hydrogenate them. So our microbes are hydrogenating them. And if you remember, within a rumen, we have high concentrations of hydrogen. So there's adequate hydrogen around, so they are able to hydrogenate that fat and they make it saturated fat. So that's one of the reasons why our ruminants, even though they're eating plant diet, a diet consisting of plant origin, which has a lot of polyunsaturated fatty acids in it. However, when you eat steak, for instance, the beef steak is going to have a lot of saturated fat. Well, that's because of these microbes. These microbes are hydrogenating it and converting the unsaturated fatty acids to saturated, and then they are absorbing that saturated fat. In addition, because these microbes are hydrogenating, and as I mentioned in the last lecture when we described hydrogenation, we can have the formation of trans fats. So our fats within ruminants end up having more natural trans fats, and that's because that's coming from the microbes hydrogenating it. So this is where we get the conjugated linoleic acid, or CLA. So our conjugated fatty acids are coming from this hydrogenation. We also have a higher incidence of our odd chain fatty acids and our branch chain fatty acids, and this is typically what's giving the distinct odor within that animal. If you've ever eaten, for instance, lamb or goat, when you're cooking it, it has that very distinct aroma and flavor, and that's due to the type of microbes within their GI tract end up making more of these branch chain and odd chain fatty acids, which gives it its distinct flavor. And we do have within the rumen the lipolysis, or the majority of the breakdown of the lipid, is occurring within the rumen. So basically that means that the majority, because we have all this lipolysis occurring within the rumen, that results in 80 to 90% of the lipid is entering the small intestine as a non-esterified fatty acid, also referred to as a free fatty acid. I use those two terms pretty interchangeably, but the correct term is a non-esterified fatty acid, or NEFA. Our fatty acids are also converted to soaps because we have a lot of ions within the rumen. That's having those non-esterified fatty acids are better able to interact. That acid component on the carboxyl end of the fatty acid is able to interact with the ions. So we have the potassium and calcium are interact. So our potassium soaps are more readily absorbed than our calcium soaps. And then our phospholipases convert biliary lecithin to lysolecithin, and that improves saturated fatty acid solubility. So here we have a good summary of what's going on with our lipid digestion and absorption, and this is primarily with our non-ruminants. So here we have our lingual lipase is able to attach to the lipid droplet, and the large fat droplets break down into smaller ones. That lipid droplet enters the duodenum, and we have our pancreatic lipase binds to the fat droplet, and we start having the fatty acids, monoacylglycerides. They're able to enter these micelles, and we have our bile salts conjugated to that. Once we have these mixed micelles with the bile salts and the phospholipids, etc., and they're small enough, those are able to diffuse into the unstirred water area, and they're able to merge, and that's how we're going to be able to absorb our fatty acids. So we'll start off with lipid absorption. So our lipid absorption is both passively diffused, and we have facultative transport. So here we have our micelles are formed, and then they're able to merge into the unstirred water area and encounter the microvilli of the enterocyte. Once it merges here, we have our monoacylglycerides. Those passively diffuse. Right here, you can see those are able to easily enter into the cell. Our non-esterified fatty acids, or free fatty acids, are a little more challenging, so we have to account for them. If they get into the center of the phospholipid bilayer, they're not going to leave, so we need a transporter. So here we utilize the fatty acid transport protein 4, and they are able to enter in. And we also have it's a dimer with CD36. Not so important for this class. I mainly want you to know about the fatty acid transport protein 4, and then that fatty acid is able to enter in, and it quickly binds the fatty acid binding protein. So we have passive absorption, and our NEFA is coming in through our fatty acid transporter. Our monoacylglycerides are passively transported, and we have 95% of the lipids are absorbed, which is quite amazing. So once we have our non-esterified fatty acids coming through, we quickly bind it to our fatty acid binding protein. And very quickly, we also end up re-esterifying it to a triacylglyceride within the ER. And this is very rapid. It's occurring within 30 seconds of that fatty acid being absorbed, because we really want to make sure we don't form any detergents within the cell. Our rate-limiting step of moving the triglyceride from the ER is going from the ER to the Golgi. So this component here is our rate limiting. And then once we have the triglyceride go to the Golgi, we then repackage that, and that is what we refer to as a chylomicron. Our chylomicron is able to pass out of the cell into the lymphatic system. So it's a chylomicron if it goes into the lymphatics. In birds, it's going directly into the blood, because if you remember that they do not have adequately developed lymphatic system, so for them it is called a portomicron, because it's going into the portal vein. And notice here we have this lipid droplet. So these enterocytes have a significant amount of lipid storage that they are capable of also. So what is this fatty acid binding protein, and why do we need it? So our fatty acid binding protein is used to prevent unexpected detergent effects. So detergents are really bad to form. Remember, we have these non-esterified fatty acids that hydroxyl or carboxyl group is charged at the end, and it can interact with various ions. There's a lot of ions within the cytoplasm of the cell that can cause detergents and disrupt the whole cell membrane. Just think of when you are using soap to wash dishes, etc., the way that soap works and it breaks through grease. That's basically what it's going to do to your cell membranes. So it's going to envelope the fatty acid. So here you can see all this blue around. This is your fatty acid binding protein. And our fatty acid is right in the middle, which is this white part. And this red part over here is basically what is the binding site where the fatty acid is binding. So our blue is our fatty acid binding protein, our white is our fatty acid, and this is binding at the hydroxyl group to keep it from interacting. So this is preventing any detergents from forming. We have to have the fatty acid, the free fatty acid or the non-esterified fatty acid bound to this fatty acid to prevent detergents. So next we're going to talk how cholesterol and various sterols are absorbed. So first, the sterols are esterified to fatty acids when they are consumed in the diet. And the main sterol we're concerned about is cholesterol. And we utilize the lipid esterase to cleave the fatty acid from cholesterol. And then the fatty acid is hydrolyzed first, and then we are able to absorb the cholesterol after. So cholesterol is absorbed through facilitated diffusion, and it has its transporter is NPC1L1. Not really all that critical to know, but this is how we're controlling the amount of cholesterol that we absorb. So we're able to upregulate and downregulate this transporter based off of our diet. So if we have a significant amount of cholesterol in our diet, we're going to downregulate the expression of NPC1L1. And the reason why we have to have the fatty acid hydrolyzed first is because the cholesterol cannot fit through the NPC1L1 with the fatty acid conjugated to it. Now here you see these other compounds that I have listed: cytosterol, camphosterol, stigmasterol. These are from plants over here. So all of these ones are from plants. So only animals make cholesterol. And this is, as I mentioned before, because cholesterol is very critical to maintaining the structure of the cell membrane, and it's because of cholesterol that we do not need cell walls like many bacteria and like plants that have to. So we regulate our cholesterol or sterol absorption pretty closely. And the way we do this is with various nuclear hormone receptors. So the first hormone receptor we're going to discuss is FXR. So this is specific for cholesterol, and these nuclear hormone receptors, they always act as a dimer. They're always conjugated to another nuclear hormone receptor. So this first one, which is in the liver, is FXR, RXR. So our FXR is farasinoid X receptor, and our RXR is retinoid X receptor. And this comes together in the liver, and you end up utilizing this, and this causes a downregulation of our bile acids, our bile salts. So if we decrease the amount of bile salts, we're going to decrease our solubilization of our cholesterol. So therefore, if we're decreasing our solubilization, we are decreasing the likelihood or the amount that can be absorbed. So we have decreased absorption here. So the smaller arrow is indicating right here, this is indicating lower absorption. So if we decrease our sterol solubilization, we're going to increase the amount of it being eliminated. Out of the cholesterol that is absorbed, we have it's converted to cholesterol esters and put in chylomicrons, and it's brought to the liver, and we're going to end up keeping this cycle going. In addition, within the cell, within the enterocyte, we have these ABC transporters. That ABC stands for ATP binding cassette transporter, and that increases the excretion of cholesterol and various sterols into the intestinal lumen. So here, yes, we're going to have some absorbed, but we're also going to increase using LXR, dimerized to RXR. So that's going to increase the amount of these ABC transporters that's going to be kicking the cholesterol back out into the lumen so it can be excreted. Our ACAT2, which is acyl-CoA cholesterol acyl transferase 2, that's what's converting our cholesterol to cholesterol esters within the enterocyte and allowing that to go into the chylomicron. And if you have a deficiency in our ACAT2, it actually causes diet-induced hypercholesterolemia. And if you know anybody that has significant amount of gallstones that have formed, this is usually due to this deficiency. Next, we're going to change gears and discuss lipid distribution within the blood. And with the main way is we discuss this is with lipoproteins. So what are lipoproteins? Lipoproteins are the proteins that are incorporated into the lipid droplet. So we have various different types of lipoproteins, and that is what the type of lipoprotein is distinguishing the different transporters of lipid throughout your blood. These proteins are amphipathic, so they are both hydrophilic and hydrophobic, and they carry dietary lipids and synthesized lipids. And we have 9 different proteins that make up these lipoproteins. So the structure of the lipoprotein, so here we have a picture on the right-hand side of a fat transporter within your blood. So what's making this, we have a shell of our phospholipids. So you can see all of our phospholipids. The phosphate group is purple. The fatty acids conjugated to it are yellow. Phosphate group is pointed out. The fatty acids are pointed in. So we also have, so these lipoproteins are located within this phospholipid layer, and that's because we have a hydrophilic region which points out, and then we have our hydrophobic region that points in. So our outside of these proteins are going to be hydrophilic, our inside is going to be hydrophobic. So on the inside of this is where we have our cholesterol esters, our triglycerides, and any fat-soluble components. So what other fat-soluble components would be on the inside? So this would include our fat-soluble vitamins, for instance, A, D, E, and K, and any drugs that we consume. So a lot of drug companies will utilize this. If you have any long-lasting drugs that you're taking orally, they've synthesized them to be more hydrophobic. So the reason why they are longer lasting or have slow release is because they are in the middle of this hydrophobic region. And our lipoproteins transporters, they come in a variety of sizes. So we have very large ones, which are our chylomicrons, and then we have very small ones, which is our HDL. So what are our lipoprotein classes? And this is one of the things that typically when you hear in the media or reading articles, newspaper articles or magazines that are discussing health, they discuss lipoproteins, but they misclassify it and call it cholesterol. So our lipoproteins, they transport cholesterol but are not cholesterol themselves. So our first class is our chylomicron, and it is a chylomicron in mammals. In our avian species, it's referred to as a portomicron, and that's because our poultry and other avian species have a less developed lymphatic system, so it does not go there. These lipid components, transporters from the enterocyte, are not going into the lymphatics, they're going into the blood. It's porto because it's going into the portal vein. These are the largest by far out of all the lipoprotein classes. They have the lowest density. So when we're talking about density, we're talking about the amount of protein, not the amount of fat, not the amount of cholesterol. It's specifically looking at the concentration of these proteins within the phospholipid layer. It also has the highest percentage of triglycerides, and this is what's carrying the triglycerides from the intestine to the liver, the skeletal muscle, and the adipose tissue. Our next classification is our very low density lipoprotein. It is the second largest lipoprotein. It also has the second largest percentage of triglycerides. So any of the triglycerides that our liver has synthesized, if you're eating too many carbohydrates and you need to convert that glucose into a more easily, readily stored form, we're going to use that VLDL to transport those synthesized triglycerides that originally came from too many carbohydrates to our adipose tissue. Our next class is our intermediate density lipoproteins, and these are not detectable in fasted animals, and they really don't last too long even if you have been adequately fed. They're intermediate, so they don't they're not too easy to find. Basically, when the intermediate, because as we drop off the triglycerides from our VLDL, as the amount of triglycerides gets smaller and smaller, our VLDL turns into an IDL. And then we have our next classification, which is LDL, or low density lipoprotein. And this is referred to as cholesterol because it has the highest concentration of cholesterol esters out of all the lipoprotein classes. And because of this, it's referred to as bad cholesterol, but it's not a cholesterol, it's just a carrier of cholesterol. And it delivers all the cholesterol from both the diet and what your liver has synthesized to the tissues. And the reason why this has a bad association to it is because this is what is correlated with higher risk of atherosclerosis, or the development of plaque in your arteries. So basically, if you have these high level of LDLs, you have a higher chance of these LDLs to become oxidized by free radicals that are within your system. Once that LDL becomes oxidized, it is no longer looks familiar to our immune cells, specifically our macrophages, and then our macrophage will consume this, and this oxidized LDL actually ends up almost like brainwashing the macrophage. It alters the function of the macrophage, and it becomes what we refer to as a foam cell. And so it is unable to clear the oxidized LDL. In addition, it signals more macrophages and to that location, and those macrophages will end up becoming foam cells. So then you get a buildup of plaque. So buildup of those foam cells becomes plaque. So that's why it's referred to as bad cholesterol. And then our HDL, or our high density lipoproteins, this has the highest density of protein within that phospholipid compartment. So it has a low lipid, high protein ratio. And this is what we deem good cholesterol. So why is it good? Because it is collecting any excess phospholipids, any cholesterol, any triglycerides from the tissues, and it carries it to the liver. And that's so the liver can then package those up and send them into the back to the intestine, and they'll be excreted into the intestine, and that's how we clear a lot of our excess cholesterol, for instance. So this is why it's referred to as good cholesterol, because it is clearing any excess cholesterol. So this is just a couple pictures showing the difference in size of the lipoprotein classes. So you could see our chylomicrons or portomicrons are by far the biggest, followed by our very low density lipoproteins, then our intermediate, and our intermediate is just as our VLDLs drop off our triglycerides, they become intermediate. And then we have our low density lipoproteins, our lipoprotein A here, so this would be our oxidized one. So that's what that's indicating. And then our high density lipoproteins are the smallest. And you can see here by the classification, you could look at how the amount of triglycerides and protein varies. So here we could see our chylomicron has the most amount of triglycerides and the least amount of protein, where our VLDL is mainly triglycerides, but we have much more of our phospholipid cholesterol and a little more protein. And our HDL has the greatest amount of protein than equal amounts of cholesterol, phospholipids, smallest amounts of triglycerides. So the smaller the lipoprotein, the greater the protein content. So here we have a table of our lipoprotein compositions, and you could see we have all of them except the IDL, basically because the IDL is similar to the VLDL. So first we have our chylomicrons. It's coming from the intestine. You can see the density here. It has the lowest density, and remember, density is measuring the amount of protein, so lowest density because of the lowest amount of protein, highest amount of lipid, and its main lipid component is triglycerides. And one of the ways that we can identify these different lipoproteins is based off of the type of protein that is within the phospholipid. So those specific proteins are referred to as apolipoproteins. So our chylomicrons have A, B, C, and E. And then we have our VLDL, which is coming from our liver. Again, very low density, and that's because a low amount of protein, higher amount of lipids, and that main lipid component is our triglycerides. And you can see it has B and C, so it drops the A and E. Next, we have our LDL, and our LDL is basically, as we have our VLDL become smaller and smaller and smaller as it drops off the triglycerides at the various tissues, it becomes a low density lipoprotein because we're removing more and more of the lipid, so then we end up increasing the amount of protein within it. So our source is our VLDL. Our main lipid component is cholesterol because we've dropped off all the triglycerides, so now we're just transporting that cholesterol around to the tissues. So as tissues need more cholesterol, it will take it from the LDL. And the main lipoprotein for this, or the apolipoprotein, is B. And then our next category is our HDL, and this is coming from all of our tissues. All of our tissues are basically synthesizing HDL, and so our main sources are going to be our liver, intestines. They can also be remnants of the VLDL and chylomicrons. You could see highest percentage of protein, and the lipid components within it is mainly going to be phospholipid and cholesterol, and it's similar to chylomicrons, has all the same apolipoproteins. So the way that we distinguish our chylomicrons from our HDL is based off of the size. And then we have, to compare to a protein, albumin is our main circulating protein in our blood. So it obviously has a very high density because it's all protein, and that's how our non-esterified fatty acids get transported. They bind to our albumin. So this is a summary of how our lipid is transported. So here you can see it starts up here. This is part of the intestine, your esophagus. You have your dietary fat come down. We get the formation of the micelles here. Our triglycerides, etc., are all absorbed. We have the formation of our chylomicrons. You can see our chylomicrons are entering the lymphatics. They go to the blood, and then they go to all the tissues. So all the blue is what is being coming from your diet. So this is our fats are going to the muscle and to our adipose from our chylomicron. And then we have our remnants. Our remnants over here are going to the liver. So our liver is taking our chylomicron remnants and repurposing those, and now we're going to have any of the triglycerides we synthesized are repackaged, and they are put as VLDL. So our VLDL, so now we've switched from blue to red. Our VLDL is what's circulating that our liver has made. And our VLDL enters the blood as it circulates, so it's dropping off some fat at the adipose. It then becomes an IDL, intermediate density lipoprotein. Some of that goes back to the liver. The vast majority of it continues on as an LDL. Our LDL is going to our extrahepatic cells, so that means non-liver cells. And when our LDL gets small enough, it becomes an LDL remnant, and it has a receptor that the liver picks it up and clears it. And then our extrahepatic cells, they are making the HDL over here, so it has excess cholesterol and phospholipids, and it is being taken to the liver. And all that extra cholesterol is being taken to the liver, so then the liver can utilize it, make some bile salts, etc., and we can excrete it. So this is another picture of basically the same thing, a little more cartoony. So I provide them both. Maybe this one, it makes more sense to you explaining it. So the downfall to this one is it does not start out specifically with chylomicrons. It has here in the intestine saying cholesterol. Well, just know that this is coming from chylomicrons going to the liver. So your chylomicrons are carrying your cholesterol from your intestine to your liver. Your liver is then taking that cholesterol and packaging it up in the VLDL. As that VLDL goes around all your tissues, it becomes LDL. So you could see the ship gets a little smaller. Cholesterol concentration gets more, so the LDL is delivering cholesterol to all the other cells. All these houses are little cells. It has to pass the plaque monster, so this would only happen if it becomes oxidized. Then the plaque monster will eat it. If it's not oxidized, then it's fine. And then we have our cells here that have excess cholesterol. Um, they are going to be putting their excess cholesterol on the HDL, and that comes around, and we have our HDL can transfer cholesterol to the VLDL, and it basically all gets brought back to the liver. So whichever picture works better for you, that is fine, but that's why they're both here. So what happens if we don't adequately absorb the amount of fat within our diet? So something happens within our intestines. Maybe you've had intestinal surgery and you just don't have adequate time. Maybe you have too much acetylcholine, so you have your flow rates going too fast, so you're just not able to absorb all the fat. Well, that results in steatorrhea. So steatorrhea is very similar to diarrhea, but where diarrhea is mainly composed of water, steatorrhea is due to the amount of fat within the diarrhea. So it occurs in a variety of various disease states. You can have viral diarrhea if you have a diseased gallbladder to where you're not releasing adequate bile. You're going to result in steatorrhea. So basically, the downfall to steatorrhea over diarrhea is very greasy. It's very odoriferous, and usually you can tell because there's decreased specific gravity, there's less protein, so the stool floats. Usually you get abdominal extension and flatulence. If you remember that our bacteria within our hindgut do not really like to have a lot of fat, especially polyunsaturated fats are actually toxic to them. We will get fat fermentation. They're going to have to adjust to that, and we'll have, that's what's causing the extension and flatulence. And when we have the fat malabsorption, we also have a secondary deficiencies in our fat-soluble vitamins, because we need our fat-soluble vitamins to be absorbed in with our triglycerides, etc. So they actually are not able to be absorbed then. So we have secondary vitamin deficiency. So how do we cope with steatorrhea? Well, this is where we have the ileal brake. So our L cells in our distal ileum, they sense the presence of increased lipids. And these L cells are what's going to cause the release of peptide YY or PYY. And if you remember that the majority of the PYY is in the distal ileum, and how it acts, so it acts in both a paracrine and endocrine fashion. The endocrine fashion is it's going to signal to the stomach in slow gastric emptying. We're also going to decrease our intestinal motility, so we're going to decrease our flow rate. And that's going to help our enterocytes absorb the nutrients. So our PYY is the main thing that is compensating and correcting malabsorption of lipids, because it's seeing all this fat. If you remember, those cells have little microvilli in them too, receptors. They're going to pick up that fat or notice that fat, and that's going to signal the release of the PYY, because if you remember how all of our animals have evolved, that we evolved where energy was not so prevalent in our diet. So we wanted to make sure we want to be very efficient when we do get any lipid, that we're very good at absorbing it. So we're going to slow down our GI tract for it. And this also is the same thing if you overconsume with your fat, why you're going to end up having an increase, because the more fat that's reaching the distal ileum, we really slow down our flow rate. So therefore our passage rate, we're going to end up having fewer bowel movements. So fewer bowel movements means that we're going to have an increased exposure to those secondary bile acids, which, if you remember, are carcinogenic. We're going to have an increased incidence or a higher risk of colon cancer. So that's how it all comes together.